Yeah, there was a puzzle, an ongoing puzzle with the Cranachia and especially Sulfolobus, that they tend to have complex CRISPR-Cas systems. So they carry 2 or 3 different types of CRISPR-Cas system, and they have very large CRISPR loci. But they are also hosts for many viruses. So in fact, we use Sulfolobus solitarius to propagate viruses in the laboratory before we were aware of the CRISPR-Cas system. And so retrospectively, we assumed that the CRISPR-Cas systems were not very effective, that they sort of kept down the level of the virus, but they didn't destroy the virus or didn't remove the virus. It would seem now that with the very recent work on the anti-CRISPR proteins in Sulfolobus and many homologs thereof, that there was a constant battle going on between the viruses and the CRISPR-Cas systems and the 2 or 3 different CRISPR-Cas systems. And there was some sort of equilibrium where the viruses could propagate at a lower level. So maybe the complexity resulted from this ongoing warfare between the viruses and with their anti-CRISPR proteins, multiple anti-CRISPR proteins, and the host trying to get bigger and better CRISPR-Cas systems. And so you end up with some sort of equilibrium where the viruses survive and the CRISPR-Cas immunity is very complex. One of the more interesting discoveries with the CRISPR-Cas systems was that there is the potential for interviral warfare exploiting the CRISPR-Cas system. So there was one Bicaudo virus that could co-infect Sulfolobus species together with plasmids, conjugated plasmids, or other viruses, different viruses. And the virus itself was never was resistant to the CRISPR-Cas system, but it induced CRISPR-Cas immune response against the co-infecting plasmid or virus. So one assumes that the Bicaudo virus that was not affected by the CRISPR-Cas system carries some anti-CRISPR proteins or generates some anti-CRISPR proteins. But there must also be a mechanism whereby it can trigger CRISPR-Cas adaptation against the co-infecting genetic elements, which were apparently not subjected to CRISPR-Cas when they were infected alone. So you had this possibility of the viruses exploiting the CRISPR-Cas immunity system to fight amongst themselves, and depending on which CRISPR-Cas systems the host has, if the virus has an anti-CRISPR protein against a specific type of CRISPR-Cas in the host, then it can win this battle between 2 viruses or 2 genetic elements. If it doesn't have an anti-CRISPR against that particular CRISPR-Cas in the host, then it's potentially going to lose.